Thank you, and good morning, and welcome to the ARM meeting. I'm very pleased to be here. I know that many of us, this is our first live conference in over a year and a half, and congratulations to ARM for making us feel safe and secure so that we could gather once again. So I'm going to be presenting to you today about Applied Genetic Technologies. We're an orphan ophthalmology-focused gene therapy company. Here's our forward-looking statements. And we have built a potential best-in-class late-stage product in our X-linked retinitis pigmentosa program, as well as 2 additional high-value clinical programs, and it's all built upon an integrated gene therapy program, which includes a very excellent history of developing and perfecting a manufacturing process. And we recently announced that we're leasing a build-to-suit manufacturing facility that will be operational in early 2023. So this is our product pipeline. Today I'm going to focus on those top 3 clinical programs given the time that we have, but we do have a very diversified technology pipeline with additional ophthalmology programs, 2 CNS assets, and a partnered otology program. So looking at our program for X-linked retinitis pigmentosa, this is a substantial patient population, about 20,000 patients in the US and EU, a devastating diagnosis in that these kids are diagnosed in about the kindergarten or first grade age, and they know that for the rest of their lives, their vision will get worse and worse and worse every day until they become legally blind. So when we think about that technology platform I talked to you about earlier, we really customize each portion of our product to the specific indication. And we do all the screening for the capsid and the promoter in non-human primates. I think we've heard a lot today about how there's not maintenance of specificity or tropism across species. So we think that this is really critical to do the selection in non-human primates. We also, in XLRP, have been able to work in a naturally occurring dog model, partnering with the University of Pennsylvania. We're the only people in the space that have used this naturally occurring dog model, and we think it's enabled us to really put a very good product together to give us the best chance of success in the clinic. And that clinical data supports that we did our job right in the preclinical studies, and that we have seen a 50% response rate in our XLRP patients in having improvements in visual sensitivity, which really correlates to the patient anecdotes of the improvement in their daily lives. Additionally, we've seen that in the treated eyes, we have statistically significant trends in improvements in visual acuity compared to the untreated eye. No other group working in the space has been able to show this supportive data of improvements in visual acuity. And significantly, most recently, about a week and a half ago, one of our investigators, Paul Yang, has done a careful analysis of retinal structure in these patients. And what he was able to present was 2 very important conclusions. One, poor retinal structure predicts, at baseline, predicts patients who will not respond to treatment. And number 2, that the improvement in retinal structure over time correlates to the improvement in visual sensitivity over time. And so we think these 3 findings, improvement in visual sensitivity, statistically significant trends and improvements in visual acuity, and improvements in retinal structure show that our product has a positive effect on the patients. And so we're very excited to be moving forward into a registration-facing trial. It'll be 2 treatment arms, a low dose and a high dose, and a fully untreated control arm, 60 patients. And importantly to this study, we're adding an additional test. It is a mobile navigation challenge, which is very similar to the maze test that was used by Spark for the approval of Luxturna. So we're very excited about this data. And the important thing off this slide, which it maybe looks a little busy, is all of the forward data readouts that we're going to have over the next 18 months. First of all, there will be 3 months from our Skyline trial data, which will be the first look at whether that maze data correlates to the improvements in visual sensitivity. Then we'll have 6-month data from our VISTA trial in 2022, as well as 12-month data from the Skyline trial, and also 24-month data from our phase 1/2. So very data-rich environment going forward. We would consider success to be that the Skyline data and the VISTA data matched our phase 1/2 and showed that robust 50% response rate, and further increased success would be if that maze data correlated with the visual sensitivity. Our next program is Anachromatopsia, another orphan ophthalmology indication. It has a slightly larger patient population, about 28,000 patients in the US and EU. There are different genes that cause achromatopsia, though. About 50% of achromatopsia patients have defects in a B3 gene, and about 25% of patients have defects in the A3 gene. So we ran 2 parallel trials in achromatopsia. It's a very different disease than XLRP. These patients are stable over time, but they're born legally blind, very sensitive to light. They could never be standing where I'm standing right now. And as well as they see only in black and white and shades of gray. The color of blindness they don't really care about. It's this light sensitivity and being legally blind that are the significant effects for them. So our latest data from the phase 1/2 trials of achromatopsia B3 and A3 really support moving forward the B3 program into late-stage development. So we are currently preparing our end of phase 2 submission for the B3 program to be able to move that into phase 3 development. The A3 program came as a little bit of a surprise to us in that the data there was not supportive at this time of moving forward. And after very significant genetic analysis with our key opinion leaders, we discovered that the A3 mutation produces a protein, a non-functional protein, where the B3 mutation produces no protein. So we really had a cleaner slate for treatment in the B3 than we did for A3. So we're stepping back and really exploring the A3 program in more detail while we're quickly moving B3 forward. So that data that we produced for the B3 program saw, again, improvements in visual sensitivity in patients over time. And also, when we look at a specific example, I'm just in the interest of time showing only one example, where we saw that improvement in visual sensitivity perfectly correlated to the area that was treated. So that gave us real confidence that this was a true result. On top of that, in many of the patients, we were able to get multifocal ERGs, which is a measure of electrical functioning. And again, the improvements we saw in this electrical functioning were right where we placed the product and right where we saw increases in visual sensitivity. So this all taken together gave us great confidence to move forward. Additionally, one of the biggest things these patients complain about and affects their quality of life is visual discomfort, light discomfort. And so again, we saw improvements in their light discomfort. Now, all of you who are really good at reading graphs, and I'm sure many of you are, can see that the treated and the untreated eye are both moving up in these patients that responded. And you might say, well, why is the untreated eye improving? That's because light discomfort is really something recognized by the visual cortex. And there's something called cortical adaptation, as well as neuronal crosstalk, because the neurons that go from the 2 different eyes actually cross each other before they go to the visual cortex. And for those of you really interested in exploring this further, we do have a presentation on our website from our research day by Dr. Joe Carroll, who really goes through very carefully how this cortical adaptation can happen. One of the things that I like to tell people is to think about if you cover one of your eyes and shine a light into your other eye, both your eyes will blink. You will not, even though the covered eye has not seen or been exposed to the light, both of them will blink. So that's a very simple experiment you can do. So for B3, again, we have a data-rich environment going forward. At the end of this year, we will have the 3-month data on our pediatric patient population, where we dose children between the ages of 4 and 8, and so we'll have 3-month data on those patients. And then in 2022, we'll have the end of phase 2 feedback on the B3 program from the FDA, so that we'll be able to guide the forward-looking pivotal trial for B3. Now, one thing I did want to mention that wasn't spelled out on these slides is that we did experience in the very high dose in the youngest patients. So this is the highest dose group that we treated in 4 to 8-year-old patients. We did experience a safety signal. Unfortunately, these patients did experience severe inflammation in their eyes. We had to increase the steroid dose, and I'm very happy to report these patients are all doing quite well at this point. But this dose is not a dose we would plan to move forward. And it's important to recognize that in our XLRP trial, our B3 trial, and our A3 trial, we treated across all those same dose ranges in teenagers and adults and didn't see any signal like this at all. It is really limited to the very highest dose in the very youngest patients. And the next dose group down, which we identify as dose group 5, we saw no issues in even those very young patients, 4 to 8 years old, with the dose group 5. So we feel very confident that overall we still have a very well-tolerated product and that we have reached the maximum tolerated dose for 4 to 8-year-olds. So briefly, I wanted to talk about our partnered program in optogenetics. So we have a partnership with a company in New York City called Bionic Sight. They have recently announced phase 1/2 data where we are providing kind of the vector technology. They're providing the optogenetic technology and specifically a very unique wearable device that recodes incoming light waves so that the brain can process them better, whereas we're providing the vector that transduces retinal ganglion cells with a light-sensitive protein that was licensed out at MIT. It has a much wider frequency range that it operates on and finer details. And they recently showed very encouraging data about the safety of the program, as well as these completely blind patients, when they entered to the trial, are now able to recognize light, they're able to recognize objects, and they're able to recognize motion. And we feel that this is first data, it's very encouraging data and best in class. So just in summary, this is our pipeline. I did talk today about the XLRP program, the Anachromatopsia program, and all of the data readouts we're going to have over the next 18 months. Our partnered program in RP, the Optogenetics program with Bionic Sight, they will be releasing data on the high-dose patients in their phase 1/2. And I wasn't able to really spend too much time on our dry AMD preclinical program, which we will be in IND-enabled tox studies in 22, as well as our 2 CNS programs that we're actively looking to partner. The FTD program is in toxicology studies now, and then we have a partnered program with Autonomy here in San Diego for hearing loss caused by GJB2, which is the largest cause of congenital hearing loss. And so we feel very proud about our diversified pipeline, about the number of programs that we have, and the data that we're going to be able to generate over the next several months to be able to make the lives of patients better. So I thank you for your attention today, and I hope you enjoy the rest of the conference.